Yeah, first off, thank you all to the organizers for having me here. This is really, really exciting. As some of you know, I actually did my PhD here. I listened to many talks in this auditorium, so it's a super huge and super cool honor to sort of be able to come back here and talk about my current work. So this is really a lot of fun. Okay, so as we know, right, as sort of Matthias alluded to, when we move through the world, so as we're sort of engaged in the world, as we're forming sensory perceptions, this involves the action of not just individual neurons, but of large-scale networks that act cooperatively and in complex fashion to process that information, right? So you can see this in the visual cortex. If we record from single cells, it's a single neuron here, very selective to orientation. We change the angle of the bar of light, the response goes away. And in the visual cortex of animals like humans, non-human primates, carnivores like cats and ferrets, these neurons are organized into these columnar structures where nearby neurons have similar tuning properties. So this is now two-photon imaging showing a cortical column. We see neurons color-coded by their orientation preference are all clustered together. If we zoom out a little bit further, we can see this columnar structure extends across millimeters. So now at each point in retinotopic space, we have these columnar representations of different orientations. And if we consider the neurons that all sort of care about the same orientation, we find that they are coupled together in these large-scale networks that span millimeters, right? So these neurons are preferentially coupled together. They have functional long-range connections in the mature system, and they all respond to the same thing. So we also know, and maybe for this audience I don't need to drive this point home, but I still like to do it, that development matters, right? We have a host of examples from experimental work to neurodevelopmental disorders that tell us that perturbations or disruptions early in life can have profound consequences for what happens in the system. So in order to understand how the system works, we really need to understand how it gets built. So that brings us to the topic of both many of the talks in this symposium and the thing that my lab really focuses on. So what are the mechanisms that give rise to the formation of these large-scale networks? And so in my lab, we do this work primarily in a ferret as a model system. We do this for a couple of reasons. First, they have these columnar representations that we see in humans and non-human primates. And also, there's this beautiful model for developmental accessibility. So ferrets are born relatively immature to other species. And a lot of developmental events that are really interesting for us happen postnatally. So eye openings around postnatal day 30 to 35. And so we have developmental access to time points we don't really have access to in other species. So around the time of eye opening is when we can first start to see these visually evoked orientation preference maps, right? We can get this column representation around that time. What we found, and this is work that I did when I was a postdoc, is that we actually can see these column representations, this modular structure, already is sitting there in spontaneous activity in the networks at least 10 days, if not more, prior to eye opening. So what we're seeing here, this is wide field GCamp imaging. This is a lightly anesthetized animal. And so this is like a 3 millimeter cranial window, spontaneous activity, animal is just sitting in the dark. And what we can see here, we can see these are just two spontaneous example events. And we can show that both we see this modular structure. If we look across events, we see that there are these really strong and long-range correlations across millimeters of this cortical area. Right, so what we're doing here is just showing the correlations relative to this one seed pixel here in green. We can see that there are positively and negatively correlated regions that are separated by millimeters. Okay. In addition, we know that these early correlation patterns have some predictive power for the future structure of the orientation preference map that emerges at eye opening. So these are longitudinal imaging experiments. We put a cranial window on the animal starting about 10 days prior to eye opening and followed the activity patterns over development until the eyes opened and we got visually evoked responses. And what we can see is that while there is certainly some refinement and plasticity of these correlation patterns, we also can find statistical signatures of the future orientation preference map. Now it's worth pointing out that this isn't a rigid scaffold. It's not that these early correlations are the future orientation preference map. It seems to be plasticity of both the visually evoked responses and the spontaneous activity over this period of time. But they are certainly something that seems to set at least some of the tone for the future visually evoked responses. Now, when you think about what are the mechanisms that might give rise to these activity patterns, well, we know that while in the mature system, these long-range columns are preferentially interconnected with long-range horizontal axons, those horizontal axons aren't really present yet at this point in development. Additionally, we know, and we've shown that feed-forward activity from the retina or in the LGN isn't required for these columnar or these modular activity patterns. We can silence the LGN with mucimol, we can block retinal activity with TTX, and we still see these patterns. Right, so this led us to, as Matthias mentioned, these LELI self-organization models that can generate these long-range correlations, can generate this modular activity using purely intracortical connectivity. And so the question then is there a way to test this, right? Can we actually see whether these local self-organizing mechanisms actually give rise to the modular activity in the cortex? And so just to really hammer home the point that we're trying to address here, what these systems do when they have this strong LELI interactions, they will transform unstructured input into modular structured output at a certain wavelength. So, okay, we figured, let's see if we can't test this idea. So to do that, we built a microscope that allows us to give wide field patterned optogenetic stimulation, so we can put up any pattern we want while simultaneously imaging GCamp. So the idea is we're going to do this experiment. We're going to put in unstructured optogenetic activity, and we're going to see what the cortex gives us. Okay. So we're doing this in young animals, so in this period of time when we see these modular patterns of activity, but prior to the emergence of any long-range horizontal connectivity. Okay, so when I play this movie, what we're going to see is that every time that little white bar comes on, we're illuminating the cortex uniformly, unstructured, and every time we do that, the cortex responds, we get an evoked response, but that evoked response is modular, that evoked response is structured. It happens every single time we get uniform input in and modular organized structure out. So it's just a single event shown here. When the laser is turned on, again, uniform stimulation, we see a modular pattern of activity emerge, builds up, and then goes away once we turn the laser off. Now, the interesting thing is that when we give this stimulation, right, when we drive the cortex with this uniform stimulation, every time we do it, we got a modular pattern of activity, but it wasn't the exact same pattern, right? So these are 5 successive trials. Again, uniform stimulation. What we get out is a different pattern of activity each time. And what we find is that these patterns of activity sit in a roughly low-dimensional space that looks an awful lot like spontaneous activity. And so we can compare the wavelength and the modularity of this activity. If we quantify how modular these patterns are, they look very similar to what we see in spontaneous activity. And if we look at the wavelength, we find that the wavelength of these modular patterns that we're evoking with these optostimuli are both very similar to what we see in spontaneous activity and also very narrowly distributed. So again, if you remember the prior talk, these LELI networks or these LELI interactions tend to constrain activity to specific frequency bands, right? We can sort of highlight that point here, repeating a figure that Matthias showed, that these networks tend to amplify activity at certain points. So what that means is that if we use our patterned optogenetic stimuli to deliver inputs that, again, that now are not uniform but actually have input structure to them, we can stimulate with patterns with either narrow wavelength bandpass noise or large wavelength bandpass noise. And if we give these inputs that don't match the wavelength, what should happen is that the activity pattern should be pushed towards the characteristic wavelength of the network, the one that the network likes the best, the one that is consistent with the length scale of these LELI interactions, right? And we can see that, or we should see that, in individual events, right? So on a trial basis, we should see the activity patterns pushed very close to the input characteristic wavelength. And maybe in mean activity, if we average over a bunch of trials, we can see these signatures of some input-driven activity that's, again, pushed towards this characteristic wavelength. So that's in a model. But what happens when we do this in vivo? So here are stimuli. We have patterns of activity. This is random bandpass noise. All we're doing is changing the bandpass filter to produce input patterns of different wavelengths. And we can drive activity to these patterns. What we can see here is that in individual events, it doesn't really seem that the output resulting activity patterns cares very much about the input wavelength. We can see that quantified here across a whole bunch of different wavelengths. The dashed line here is input matching output, so that would be what we'd expect if the input was driving things perfectly. This blue line here is the wavelength of spontaneous activity. So what we see, so if we take sort of spontaneous activity as the cortex telling us about what its intrinsic wavelength is, what patterns of activity it likes to produce, what we're seeing is exactly what we predicted in our models, that driving the cortex with inputs that don't match this wavelength, the activity is pushed towards this characteristic wavelength on an individual trial. Now, if we look at, take an average and we look across all of the trials we presented, we can actually start to see a little bit of this wavelength effect, right? We can see that we're in the average, we're seeing responses that are at a narrower wavelength if we have a narrow wavelength stimulus, and at a larger wavelength if we have a larger wavelength stimulus. We can actually see that response here, and again, it is shifted away from the input line towards the characteristic wavelength of the network. So what this argues is that these LELI mechanisms can drive the formation of modular activity in the developing cortex. Okay. Now, as Matthias pointed out, this idea that heterogeneity in these networks acts as symmetry breaking force and serves to drive the network into a lower dimensional set of states, and so therefore constrains the activity patterns the network can produce. So as Matthias mentioned, there are many ways to introduce heterogeneity or symmetry breaking into these networks. You can take these LELI interactions and distort them. You can add noise. The main point here is that anything that breaks the symmetry in the network drives dimensionality down and constrains or favors certain patterns of activity over others. And so can we see some evidence of that in vivo? So in one set of experiments, I don't have time to talk too much about, we performed imaging with a slightly faster sensor and at a faster rate. So what we're showing here is a single spontaneous event, image now at 50 hertz, so these frames are 20 milliseconds apart. We did a little deconvolution to try to pull out some of the calcium signatures that might blur these things out. What you can see is that there's some spatial temporal dynamics to this event, right? It sort of starts in maybe this spot over here and kind of spreads, and we see this modular pattern emerge over time. Now, what's wild here is that when Luna did these experiments, they imaged the cortex repeatedly over several hours, so put the animal on the table and image for several hours. And what they found was that they could find events that had the same spatial temporal structure repeatedly across hours. So this is one on the left, 8 minutes into the recording, about an hour later. This one's now over 2 hours later. And we see this many times, right? And this is just one example pattern that we see. There are many of these different patterns, again, roughly low dimensional. The point here is that there seems to be these sort of characteristic spatial temporal patterns to these events. They're not random. They're not occupying the full potential space of all possible patterns. They live in a constrained regime consistent with this idea that we have symmetry breaking heterogeneity in these networks that is pushing things into this lower dimensional set of states. So one implication of this is that if certain patterns of activity are favored over others, if the network has intrinsic connectivity that biases the patterns of activity that it likes to produce, then inputs that align with those patterns should be preferentially amplified by the network. Right? And this is sort of a point that was made earlier, but it's the idea that in these networks that are strongly recurrent dominated, this intrinsic connectivity is a real driving force for what activity patterns the network should produce. And so the question is can we test this idea? And so we're going to use the same optogenetic microscope, the same approach, and to stimulate the cortex now with patterns that don't vary by wavelength, but instead stimulate the cortex with patterns that either match or don't match the intrinsic connectivity. And so how do we do that? Well, what we're going to do is we're going to use spontaneous activity to give us some best guess estimate of what patterns the cortex likes to produce. And then we can design stimuli that are based upon spontaneous activity. So we image spontaneous activity, we compute some correlations, we draw stimuli that are in regions that are positively correlated, and then we can create stimuli that are random bandpass noise that match the spatial wavelength of spontaneous activity. So we're still within this wavelength band of what the cortex likes to produce, but we have patterns that are random. All right. And so when we did these experiments, this is an example here. Every one of these stimuli, the little red circles that come up, that's where the stimulus was presented. This was a stimulus that was endogenous, that sort of matched spontaneous activity. We get reliable responses that tend to correspond fairly well with the input pattern of activity that we give it. In contrast, this isn't exactly what we see when you do random stimuli. So top row here is an endogenous stimulus, one based on spontaneous activity. What we're showing is a mean response in 3 individual trials. And what we show here is just the contours outlining the active domains on the individual trials. As you saw in the movie, and you can see here on this figure, that those contours, those domains tend to fall in the same spot repeatedly over time, right? So each trial we tend to evoke a pattern that is fairly similar and fairly similar to the input pattern. When you stimulate with random bandpass noise, we still evoke responses. We reliably get responses, but the responses themselves tend to vary more across trials. You can see that in the individual responses here. You can see that in the contours here. They tend to be overlapping and not confined to the same spot.
And the amplitude is just a square pulse?
Yep. So for this stimulus here is a 1-second presentation of this pattern. And that produces very reliable responses in our hands.
So it's a pulse response?
Yeah. Just a square step function, 1 second on, 10 milliwatts or whatever. But yeah. So it reliably produces a response. We're not doing any pulsing or anything like that. Okay. And so when we quantify, when we quantify these results, basically this is what we just showed, which is that if we look at the trial-to-trial correlations, we see significantly higher trial-to-trial correlations, so more reliable responses, more consistent patterns of activity, if we're presenting endogenous stimuli based upon spontaneous activity, things the cortex likes to produce, versus random noise or bandpass filter noise. Likewise, if we look at how similar the response is to its input, right? How well does the evoked response match the input pattern we give? We see higher levels of similarity to the stimulus for input patterns that are derived in spontaneous activity. So in other words, if we give the cortex an input that it likes, it just responds reliably, and it responds with a pattern that matches the input. Okay. We can also look at this over time. So now, instead of giving 1-second stimuli, we're giving a sustained 5-second stimulus. And what we see here is something that sort of matches these ideas as well, that if we present this stimulus for a long period of time, we can evoke a response, but that response tends to wander a little bit. It doesn't always stay exactly the same. The cortex starts to change its pattern of activity over time. And what we find is that if we look at the intra-trial stability, so how similar the pattern is at different successive points in the trial, we see greater stability over time for these longer duration stimuli for patterns that are endogenous, patterns that are based on spontaneous activity, than for patterns that are random. And again, we can also see that in the similarity to stimulus, that difference is maintained, that difference is maintained over the duration of this 5-second long stimulation. Okay. So what this says, what sort of the thing we're building towards here is this idea then that patterns that match the recurrent connectivity, that match the spontaneous activity, should be the ones most amplified. Okay, so can we test this explicitly? Can we actually take patterns that match spontaneous activity to varying degrees, right? What we've done previously is we've taken patterns that we sort of tried to make match as well as we could. We took, measured spontaneous activity, took some correlation patterns, drew some stimuli, and we took some patterns that don't match very well at all, some random noise, right? But if this principle is actually true, then the strength of amplification should depend on how well any given pattern matches the inputs. So there's a way to do this relatively explicitly. What we actually did then is we looked at the principal components of spontaneous activity, which we know by definition match spontaneous activity to decreasing amounts as we move down the principal components. And we can design stimuli based upon these PCs. So here we're just showing stimuli in response to principal components 1 and 2, spontaneous activity. And we can see evoked responses and individual trial responses. When you look at all of these principal components, here we're looking at we simulated PCs from 1 to 10, we see that the lower rank principal components or lower order principal components, like principal component 1 and 2, the ones that explain the most variance in spontaneous activity, these are the components that produce output responses that are most similar to the input and are most reliable trial to trial. So it sort of confirms that idea that says that things that match well are the ones that are most amplified. Now, we can also do this if we look at all of the stimuli we presented, right? So by chance, some of the random bandpass noise stimuli might match spontaneous activity a little bit more than others. By chance or experiment or variability, some of the correlation patterns that we measured might align better or worse than others. And so what we did here is we're pooling all of the stimuli we presented across all of our animals. And in every case, what we're doing is computing how well that individual stimulus aligns to spontaneous activity, to align to the principal components of spontaneous activity. And what we find is that across all of these stimuli, so even the ones that are the bandpass noise stimuli, to the correlation stimuli, or the spontaneous activity-based stimuli, everything we drew, that the alignment with the spontaneous principal components is very predictive of the level of amplification. It predicts the trial-to-trial correlations and the similarity to stimulus. For the subset of experiments where we did these longer duration stimuli, it also predicts the intertrial stability. So what this says is that we see this millimeter scale selective amplification, right? We see these patterns, these input patterns that are, again, spanning multiple millimeters that are selectively amplified in these early cortical networks. And we see that patterns that match spontaneous activity, that are best aligned with spontaneous activity, produce the most reliable and the most stable outputs. What this also says is that if we compare this with sort of our understanding of how these recurrent networks work, that spontaneous activity can then be effectively used to estimate the dominant modes of the underlying recurrent connectivity. That's sort of why this analysis that we just showed works, right? That we know that patterns that align with underlying connectivity should be selectively amplified, and we show that the patterns that are best amplified are the ones that match spontaneous activity the most. So what this suggests is it provides a potential mechanism over developmental time for the effective creation of these orientation preference maps. That is, feed-forward inputs that happen to align with the underlying recurrent circuitry, we selectively stabilized, selectively amplified, potentially engaging in plasticity mechanisms, leading to the emergence of these stable and persistent evoked representations that we see at eye opening and beyond. Okay. So the title of the talk was suggesting that this is a common mechanism. And so what we've done so far, we spent a lot of time talking about the visual cortex, talking about how the evidence that we found that argues strongly that these LELI interactions give rise to modular activity and selectively enhance certain patterns of activity that match over others when those inputs are aligned with the current connectivity. Now, we know, though, that the cortex is highly varied. Different parts of the brain care about very different things, right? So we've been spending a lot of our time out here in visual cortex. We also have regions of the brain that are responding to auditory stimuli, sensory stimuli, more complex, higher order things, perhaps hunting, prey avoidance, all kinds of goal-directed things. The question is, are these modular patterns of activity, are these principles that we see operating in visual cortex, are they something that is universal? Are they something that is common, that is shared across cortical areas during early development? And so in a series of experiments, we tried to look at this idea. And so what I'm showing you here is spontaneous activity in primary visual cortex in an animal about P21 to 24. So it's a lightly anesthetized animal sitting in the dark, no stimuli. This is what we've seen before. These are modular patterns of spontaneous activity we see in V1. Okay, well, we can also look in auditory cortex. We see something fairly similar. We see modular patterns of activity spontaneously emerging. Again, this animal sitting in the dark, lightly anesthetized, no overt stimuli. And in somatosensory cortex, we see something that, again, looks very similar. So these are three sensory areas in early development that are all showing what look like fairly similar patterns of modular activity. We can also look in association areas, this is posterior parietal cortex and prefrontal cortex. And basically, everywhere we look, we're seeing modular patterns of activity that look fairly similar in their organization. And so we can see that here. These are just individual events taken from these different areas. We can see in all cases, we see these modular patterns of activity. If we compute correlations across events, we see a fairly similar structure, right? Across all of these areas, we're seeing correlations that are both fairly strong and fairly long range. So we're seeing positively and negatively correlated patches of cortex separated by millimeters. The length scale of these correlations is highly similar across all of these areas, and the structure seems very, very similar. We can also see this structure at the single cell level. So what I've shown before, that was wide field imaging. So a single photon wide field looking over millimeters of cortex. We can also zoom in and do 2-photon imaging, look at individual neurons, look at correlation patterns here. And we see in every cortical region that we've examined that we see these local clusters of highly correlated neurons that are nearby, separated by anti-correlated neurons further afield. Okay. So what this argues is that it suggests that these LELI interactions that we think give rise to these modular patterns of activity in visual cortex may actually be a universal mechanism across all cortical regions during early development. This may be sort of a default state of how the cortex is organized. It also suggests that the presence of a columnar architecture in V1 is not due to V1 specific inputs, right? So there have been a number of models that have attributed the presence of columns in V1 to functional organization in the retina or retinal specific waves. It's not saying those things might not have a role, but it's hard to argue that the same mechanisms that are sort of feed-forward driven, retinal driven, are giving rise to the same looking modular activity in such a broad range of cortical areas. Okay. In the last few minutes, I want to just touch on one sort of emerging story that we're working on in the lab, some work that is still unpublished. And so it's work that sort of hints that this sort of early common organizational state might also then be followed by a common developmental progression that might also be shared across these cortical regions. And this sort of builds on some of the ideas that were raised in the question earlier today about why we might have a columnar organization. So what we see is that if we look in early development, these correlation maps and the activity patterns that we see are highly dense. And what do I mean by that is that the correlations of a neuron with its nearby neighbors are very, very strong. So what that means is that when this neuron is active, nearly all of its neighbors are also active at the same time. And that highly dense connectivity might be really good for building functional representations during early development, but it's not really great for representing information. There's a whole lot of redundancy within these highly dense activity patterns. But interestingly, what we see is we look over time, so this is a different cohort of animals now imaged about a week later, around the time of eye opening, we can still see this modular structure. Nearby neurons are still more correlated than neurons further away, but the strength of these correlations has decreased dramatically. And if we go now a week even later, this is now a week past eye opening, modular structure is still there, but it's much weaker, and the activity is much, much less correlated. If we look at individual events, the activities generally become a lot sparser. We go from having nearly all the neurons in this patch active together, just a few cells coactive here. We can quantify that in these plots down here. We can see that the strength of local correlations drops. Notably, all of these areas still show significant local correlation. So it's not that we lost the modular structure, it just became much weaker or the correlation strength dropped. The activity becomes, as I mentioned, way sparser. So we go from these highly dense activity patterns, something that is much less dense, much sparser, and the activity becomes higher dimensional, right? So the representational capacity of these networks seems to have increased. And so the idea perhaps is that there is both a common developmental origin, these sort of modular activity patterns, perhaps followed by a common developmental maturation going from these large-scale, coarse structured representations to something more fine-scale over time. Okay, so just to sort of summarize and bring it together, what I've tried to, or hopefully convinced you of, what I've tried to make the argument here is that we have these LELI networks in early development that are transforming unstructured activity into modular patterns that have a certain wavelength and a certain subset of patterns that they prefer. Inputs that align with that connectivity are preferentially amplified within these early networks, and that this may in fact be a common mechanism that gives rise to modular patterns of activity throughout the entire cortex, and we might see common developmental mechanisms following that. Lastly, the most important slide are the folks who did the work. A large part of the optogenetics work I talked about today was led by a former graduate student in my lab, Haley Mulholland. She's now a postdoc at NIH. The imaging of the multiple areas was work led by Nate Powell. He now has a faculty position in Pennsylvania. I can't remember where. This is all part of a long-running collaboration with Matthias and two of his students, Sigrid Cheganap and Bettina Hein. Thank you all for your attention. Happy to take questions.
Thank you for your beautiful talk. I have a question. So when you show us this sort of like a stimulation, optogenetic stimulation, and you're saying, okay, if the stimulation pattern matches the structure itself, then you get an amplification. But if you think about like this sort of like structures, let's say in higher order like sensory areas or beyond, right? The input space is going to be different rather than the natural input space. That is, sending information to these cortices are different rather than the way that you're stimulating them. So how do you kind of reconcile this idea? Because, think of it that, you have a set of modular network, right? So if you don't know what's the input line basically to this set of modules, how would you go exactly and then try to reconcile what Matthias was saying?
Yeah, no, so I think that's a great point. And so there are a lot of questions wrapped up in that. And so one of them is, so we know in visual cortex that we have this columnar representation, in part because it's organized along a dimension that is intuitive, right? Intuitive enough that observant scientists notice that the edge of a slide evokes responses. I have no clue what the relevant dimensions should be in prefrontal cortex. I'd love to figure those out. That's not something I'm an expert in. So it could be that there's this early modular organization in prefrontal cortex that then gets broken down as the animal gets older. It could be something like what we see here. The activity becomes sparser and less modular, maybe as prefrontal cortex gets inputs that are different, and the scale of this amplification becomes smaller and more fine scale. It may be that that's not the case. It may be that, so we've done experiments where we've done wide field imaging at this age. I don't have the slide here, but we still see modules. We still see module activity. Now, this isn't an old animal. This is an animal like 10 days after eye opening, so, okay. But it's still there. So does it carry some representational meaning? I don't know. And so I think there's a lot of unknowns there. It could be that it breaks down. It could be that the scale of this thing changes. Some experiments we're doing in the lab now is to, instead of probing these networks at the millimeter scale, to do this at the cellular scale. So we've got a 2-photon optogenetic microscope set up, and we can ask these same questions now at the local scale. And I think that's a really way to get at these points. In recurrent dominated networks, we should see this amplification. That should be a feature of networks that are strongly recurrent dominated. It's also possible that over developmental time, the networks become less recurrent dominated, right? That we could have these networks at these early ages prior to eye opening where the current connections are the thing that, like the only thing that's really driving this activity. And it could be that these networks sort of shift to become more input driven later on. And I think there's some evidence that might be the case, but still early days.
If I may ask like one more question, basically to both Matthias and you. Basically when you start, so what you showed us here, the combination of these, both the mathematical model and the experiments, it tells us that there is this sort of like modularity in all these areas, right? But if you think about like the sort of like a landscape of, you have modularity on one end, and then you have columnarization on one end, right? So columnarization seems to disappear somehow as you go towards the higher order areas. How do you like describe this with this sort of like observation?
So I'm not certain that that's true, in that there is anatomical data from Patricia Goldman-Rakic and others. There are things that look like columnar axonal arbors in prefrontal cortex in a primate. So this is where that gets the idea that I don't know what the dimension is, right? So it's possible that there is some dimension of higher order representational goal-directed space that is contained in a modular fashion in prefrontal cortex. I don't know. One implication of some of the work that we have here is that if spontaneous activity is telling you what the network connectivity cares about, then perhaps you could use spontaneous activity as a way to back into what those representations might be. And so we have ongoing experiments in the lab. We've got some head-mounted cameras. We've got some ferrets running around in some boxes. We'll see. I don't know. But the idea is if spontaneous activity is really telling you what the cortex cares about, perhaps that can tell you what the cortex cares about. I'm not smart enough to tell you what prefrontal cortex should care about. Even my prefrontal cortex colleagues probably can't do that, but I definitely can't. So.
Hi. Really interesting. I just want to follow up with something you just said about the presynaptic arbors. I don't have a good sense of scale of your imaging, how those modular activity units, how they relate spatially to an axon arbor. Either from the, I know you said that it's not necessarily from thalamocortical projections, but intracortical projections.
Yeah, so, okay, I can answer the question partly, and partly is going to be a little unsatisfactory. The part that I can give you some answers about is what the size of these domains is, right? So these active domains are about 400 microns-ish, okay? From what we know from the anatomical data that is published, axonal arbors are sort of in that ish range, maybe a few longer projections at this time point. The reason I'm being a little hesitant here is with all of those studies that sort of inject a dye and sort of trace out an arbor, you only see the connections you see. So it's possible the dye didn't get into everything. The thinking here is that there are some connections that are maybe 500 microns to a millimeter. We don't see the really large-scale patchy connections that we see in the mature cortex. So in the mature cortex, animals say, 2 months after eye opening, what you would find, let's look at this orientation map, you'd find preferential connections from this little patch of, let's say this is vertical, to this little green patch of vertical, to this little green patch of vertical, right? These areas are preferentially connected with really long-range axons. We know that's not there, at least from any labeling that we've seen. So, clearly there is monosynaptic connections. I think in order to, from what we know, there probably aren't things that are long enough to produce monosynaptic connections across the 2 millimeter scale we see in these networks. So you need something that involves recurrent network activity to produce something like that.
So to what extent, so I do understand that your systems are nonlinear, but to what extent one can connect the 2 points that you are making, that there is a selective amplification of spontaneous modes, and the second point that spontaneous modes help you predict responses to stimuli, to ideas from linear systems like resonant frequencies and fluctuation dissipation theorem that tells you that spontaneous fluctuations are informative of responses to stimuli.
Right, and so I think part of the answer, maybe we should talk offline because I'm not sure if I can fully answer your question. But one way to think about it is what is spontaneous activity doing in the system, right? It can be thought of as some sort of representation of some sort of prior estimate of what the world should be doing. It can be thought of as some reflection of underlying connectivity. And so in these contexts, I think you can think of spontaneous activity perhaps as, we've often used it, right? And so we sort of walked into these experiments with this idea that, well, look, like, we really want to know what network structure looked like at these time points here, but we couldn't evoke a response, right? We couldn't drive the cortex in any reasonable way at these time points. So, prior to the experiments that we did a few years ago, we really didn't know what the network structure was here. And so we're often in these cases using spontaneous activity as a way to sort of tell us what the cortex knows, what it does, right? Show me something about your network structure. Now, that can have some relevance to, say, a representation. It could be, I will also say, right, like if we look at orientation preference maps and spontaneous activity in older animals, right, a whole lot of the spontaneous activity, the variance in spontaneous activity, the first two PCs, are basically the orientation preference map, right? And so there is certainly a clear sense that it does care about what the network cares about. But that's only halfway, I think, to the question.
When does the GABAergic circuitry in cortex finally mature in the ferret?
It's a great question. And I don't know finally mature. Finally mature is a different question. I think the point you're asking, though, is what is the state of the GABAergic circuitry at these time points? Do we have enough effective inhibition at these time points to generate these models? And the answer is yes. So we know, and this is work that we have published, if we put on like bicuculin or picotoxin at here P21, you'll get a seizure, right? So by this age, we have net effective inhibition, right? So if this question of thinking about is GABA excitatory during early development, if it is, it certainly isn't at this time point here. I have preliminary data that says that time point's even earlier, but again, preliminary. Now, in terms of full maturation, we're certainly like PV neuron expression, PV expression turns on around the time of eye opening. Future PV neurons are there. They're probably not fast spiking yet at these developmental time points, but there are definitely inhibitory neurons there, but they're definitely not mature until somewhere out here.
I guess I'm wondering how much it might explain your more recent kind of developmental experimental results.
Well, that's actually a very good question, because let's jump to this one. We have some preliminary, so as I said, this story is sort of unpublished, but we have some modeling work that suggests that a lot of these results, this sparsification, this decorrelation, this change in dimensionality, you can get these effects if you take a model and basically make inhibition stronger, right? So we know that during this developmental time, inhibition is continuing to mature and strengthen. And so it's a reasonable guess of a lever that might be moving the system from one state to another. And it seems to do fairly well at achieving this in models. Is that what's happening in the cortex? I don't know. I hopefully will be able to find out in the next few years. We have some experiments ongoing that are sort of directly testing the levels of inhibition and functional connectivity at various scales over this developmental time.
What is the relative timing of the arealization between primary visual area and secondary, extra striate visual areas, as well as all the, somatosensory versus the fields, the arealization, this process? That was question one. And question two is, I'm struck by how it looks like the spatial scale of these patterns is similar across V1, S1, PPC, and PFC, which, I don't know, that's partly a question to you. Is it the same? And if it's the same, that seems quite interesting and surprising, given that higher areas are supposedly in cortex much more strongly recurrently connected, and you might expect that there would be some differences in the spatial scale of the connectivity or something else.
Yeah, so to the latter point, they are. When we quantify the domain size, the wavelength of the activity, they are basically the same across all areas. And so
It
is. It is. And so what that implies is that, if we're right, and these are sort of LELI-driven interactions, that implies that the length scale of those LELI interactions is similar across these areas.
Striking.
Yeah, it didn't have to be that way, but that's what we get. To your first question, the areas so they're already specialized in some sense, in that, okay, at P21 to 24, you really can't get much in the line of evoked responses. You can maybe get some evoked somatosensory responses in the sort of around eye-opening age, so 27 to 30. That's also when the ear canals drain and the ear canals open. You can get auditory evoked responses. You can get somatosensory evoked responses. And you get the responses in the right area. So in other words, the areas are getting the right inputs. The thalamocortical connections are there. The thalamocortical connections are probably already there even at this age. You know, I think in V1 they're probably there even earlier than this in connections terminating the subplate. Response reliability is really, really hard at these ages. You can get things sometimes. So, I mean, that all seems to be happening earlier than this, and that these areas, like, we already have this sort of gradient across cortex from frontal regions to posterior. The thalamocortical connections are sort of sorting out on that sense. So if we sort of define area specialization by that, it seems like that's already there. And so I think one of the really interesting questions will be, as we sort of follow these areas and sort of get to look more and more detail at the changes over developmental time, is as these areas start to receive different thalamocortical inputs, right? PFC is getting different input than what V1 is getting. And so the question then is how do those different feed-forward inputs interact with this recurrent network structure? And does that change differently in different areas? So.
My question is, so how does the system select what patterns to amplify? If the system is built in a way that doesn't care about sensor input in the beginning, right? The emergence of these spontaneous patterns, they do not require sensor input. Why care or bother to amplify them at all in the end?
Right. So there are two ways to answer that. I'm not sure why the cortex picks certain patterns over others. You know, one way to phrase that question is, what are the potential advantages of having networks that amplify certain responses? And, you know, the typical ones are if you live in, you know, environments that are noisy or information that's uncertain, having networks that amplify certain patterns can help reduce that space and produce more actionable outcomes. I mean, you know, right, this example I use with my students sometimes, right? Like, you know, when the giant bear's chasing you, you don't get to ask him to do it 10 times and average over 10 times. You get to decide if you're going to run or not, right? And so the systems are evolved to produce decisions to answers and to try to handle noise and variability. The other point, and this is one that I don't think I made explicitly, is that, you know, when we think about these systems that produce activity at certain wavelengths, well, that's a really nice feature when you consider something like the orientation preference map in the visual cortex. So this is a map that has to develop in concert with retinotopy, right? So this part of the visual cortex sees a different part of the visual world than this part does. That spacing of retinotopic inputs determines an operating wavelength for this map that it has to be. If you make the wavelength too big, you can't see vertical orientations up here, you can't see horizontal orientations down here. The wavelength of this map has to match the retinotopy, has to be consistent with the retinotopy. So it could be that these LELI interactions are a fairly robust way to produce patterns of activity that have a certain spacing, a certain wavelength to them that can then match onto this peripheral input that's already there. So that's just another point, sort of kind of gets to that question.
Great. Let's thank our speakers from the session.
Thank you.